to today's final presentation, which will be by Dr. Maximilian Sprang. He is a bioinformatician and a junior group leader at the University Medical Center of the Johannes Gutenberg University in Mainz. He obtained his doctorate at the same university, and his research focuses on quality control of multi-omics data, generative models for DNA resequences, and AI applications in immunology. And today he's sharing with us how he aims to improve the quality of wet lab data through identifying the most fragile steps that introduce bias and distort the downstream analysis. Please tell us more about it.
Thank you for the kind introduction. So this project is basically the natural extension of my PhD thesis, which I finished last year, which was very much focused on quality control for functional genomics, especially RNA-seq. And we called it Erring rigorously. So we want to try basically to deliberately do errors or introduce differences in the wet lab to then see how they impact the genomic analysis. So just a very short introduction here on what I'm talking about. So we're on the same page. So for RNA sequencing on the top here, you see the wet lab part very, very shortened and schemed, say like that. And on the bottom, you see the bioinformatics part, again, also very shortened, right? So when we would start, obviously, with some kind of sample, this could be a cell culture, and this is also what we will be talking about today, but this can also be tissue from an animal or from patients and donors, right? We will extract the RNA from this tissue, at least if we are in bulk. If we are in a single cell, we need to get the cells first. And then we would translate them to cDNA and then throw them into a sequencing machine that looks roughly like this. And this is where my work starts normally, right? So I get the raw sequencing file. This contains millions of reads, so the sequenced counterparts of these little fragments here. And here we can already do in silico quality control. And what we normally do with these, especially in RNA sequencing, is we align or map them to a reference genome and then count them. And then this gives us a relative quantitative measure of the activity of a gene, right? And then with this, then we get a count matrix out of this. And with this count matrix, we will do clustering, we will do differential analysis, which is then done with statistical or machine learning models. And now we come to the problem here, which is a bit based in the rift between these two worlds, and also in the many, many steps that need to be done to actually arrive at the analysis. And this is often called artifacts or hidden biases, sometimes also just unwanted variation. And we are talking in general here about effects that cause differences between the samples that are not originating from the desired observation. So the desired observation here would be what we see here, right? So a group that we compare. So for example, a knockout versus a wild type, or a patient with a disease versus a healthy donor. And in work published 2024, we could show, for example, as an example for such a hidden bias, that if you have differences in quality of a sample between the groups, right? So your KO and knockout or your disease and healthy group, then this will introduce false positives in your downstream analysis. And we actually saw that this difference was, or we called this the quality imbalance. And we actually detected this quality imbalance in 30% of the data sets that we surveyed. And then we had a follow-up poster actually in ECCB the next year, where we increased that data set a lot, and we still had over 30% of this quality imbalance data sets. Perhaps a bit better known artifact is the so-called batch effect, and it's also nicer to visualize. And this is actually the difference between two batches of an experiment. So the name is quite fitting, right? You can get this by different handlers of the same experiment or different time points of the same experiment. And then you will end up with something like this here, where the signal of the experiment, which is the color here, is actually stronger than the biological signal that you are looking at. And to mitigate this, there are a lot of software solutions, there are a lot of applications out there that will help you to correct such a batch effect, and technically also other artifacts that may be hidden in the data. So this brings us to the gap of the problem here. So yes, we can correct noise, right? So we have an experiment where we do know perhaps about a hidden bias, or perhaps we can even use software to correct this bias, to find this bias, I'm sorry. And then subsequently we can correct this because we cannot use it directly, data like this. However, this only works sometimes. And the state of the art basically is to really take multiple of these softwares, run them on your experiment, and then take the output that fits best to your biological knowledge about the system, right? And obviously this is not optimal. And I believe that the reason for this is that we rarely understand where these biases actually come from. So this brings us to the idea to err rigorously, right? So we want to establish a controllable experiment or experimental environment, and then systematically and deliberately introduce technical perturbations here. And we will use software then subsequently to score the quality of the samples that come out of this. And I already mentioned that we also provided software for some of these problems, and the software is called SecuScorer. It was published in 2021 together with Steffen Albrecht. It was part of both of our theses actually. And what it does is that it is a package of machine learning models that was trained on ENCODE data. So ENCODE comes actually with quality labels, right, that we could use to train this. And then it gives you the probability of low quality P low. And this we used in subsequent work, and these two as a quantification for quality. And our experimental design would look something like this. So we would actually work in cell culture for this experiment because this gives us the most control on the system. And then introduce cell changes in the cell passage, changes in the culture density, right? Or for example, directly interfere with RNA integrity by sonification, for example. And then downstream, we take all these samples, sequence them, and then do our quality scoring, do the classical bioinformatics downstream analysis to also be able to look at the biology, right? We're not only interested in measuring quality here. And then hopefully get an understanding on how strong some of these changes are, both on the outcome of the biology as well as the quality. The expected outcomes and deliverables here are threefold. Two are immediate, and one is a long-term goal, right? So at the core here is obviously the data set itself, which would be annotated. We would know where the differences come from, right? And this information would obviously be shared with the community, and thereby this could actually be a new standard data set to test these softwares on. And then second, we could improve our own software. There are some changes that we wanted to do for some time now when we were using it. And additionally, also in such a controlled environment, we have the opportunity to test the software yet again and find options to improve it. And then finally, in the long run, a project like this could be a first step towards data-driven guidelines for generating or designing new experiments, right? So there are already a lot of guidelines around for sequencing data. Some of which I already mentioned, for example, ENCODE, right? It's a giant data set and provides very good guidelines. However, even their own quality scoring does not adhere always to these guidelines, right? And so this could be a step to make the whole process more empiric, let's say it like that. And so why does this matter for such a wide audience that the Einstein Award represents? So obviously it's about quality control and reproducibility, right? So that would be a fit, but also functional genomics are pushed more and more into translation and into the clinics. So they have been the backbone for biological research for some time. And now they are also more and more in translation, for example, in molecular tumor boards, right? And so the trust in these experiments is even more important. It's not only ourselves, the research community, that needs to trust them, but also the patients, right? And to get this trust, you need to be reproducible. And for reproducibility, you need to understand what is going on under the hood. Thanks for hearing me out. Also, a big thank you to Miguel Andrade, Julian König, Maria Mendes Lago, and Andreas von der Heid for letting me pitch this project to them and help out with some feedback. Thanks to my current employer, AG Meyer, and as already mentioned, I'm a junior group lead, so I'm building a junior group. So if you are interested in working with me, feel free to contact me.